from www.news-medical.net. What is an enzyme cofactor? By Dr. Maho Yokoyama, PhD. Reviewed by Hannah Simmons, MSc. Making life's reactions go faster. In living organisms, there are many chemical reactions taking place. These reactions have many different functions, including breaking down the food we eat and getting energy from the broken down food. To facilitate this, there are a group of proteins known as enzymes. Enzymes speed up or catalyze these chemical reactions, working with only one substrate to catalyze one reaction. Many enzymes come together to form pathways, leading to a variety of functions such as muscle contraction. Additional factors. Some enzymes require the addition of another non-protein molecule to function as an enzyme. These are known as cofactors, and without these enzymes remain within the inactive, quote, apoenzyme, end quote, forms. Once the cofactor is added, the enzyme becomes the active, quote, holoenzyme, end quote. Cofactors can either be ions, such as zinc and iron ions, or organic molecules, such as vitamins or vitamin-derived molecules. Many of these cofactors will attach near the substrate binding site to facilitate the binding of a substrate to the enzyme. Cofactors can be classified as, quote, prosthetic groups, end quote, or, quote, coenzymes, end quote, depending on how tightly they are bound to the enzyme. Coenzymes bind more loosely to the enzyme and are thus modified during the enzymatic reaction, while prosthetic groups are more tightly bound to the enzyme and are not modified. Prosthetic groups. These can be ions, such as Zn2 plus ions, used in dehydrogenase enzymes, or Fe2 plus ions, used in alkaline phosphatases. And as a brief note, if I'm not mistaken, Zn is the abbreviation for zinc, and I do know for a fact Fe is the abbreviation for iron. Just do a quick search on video. So, if we were to just go by the Gemini AI's overview, Zn2 plus is the divalent zinc cation, formed when a zinc atom loses its... Okay, so it's basically zinc. Back to the article. Molecules such as tryptophan, tryptophyllquinone, TTQ, act as a prosthetic group in reactions catalyzed by methylamine dehydrogenase. Another molecule, flavin adenine dinucleotide, FAD, can be remade during the enzymatic reaction and therefore can be considered to be a prosthetic group as its overall concentration does not change. Transferring molecules by the use of coenzymes. Some coenzymes will transfer a molecule during the enzymatic reaction. For example, coenzyme A transfers acyl groups. Thiamine pyrophosphate is another coenzyme which transfers aldehydes. Pyridoxal phosphate transfers amino groups. Biotin transfers carbon dioxide, CO2, and carbamide coenzymes transfer acyl groups. Other coenzymes such as nicotine adenine dinucleotide, NAD, coenzyme Q, and FAD transfer electrons or hydrogen atoms. From vitamins to cofactors. Vitamin-based cofactors are essential for processes such as vision, blood clotting, and hormone production. Retinol, the aldehyde version of vitamin A, are cofactors for opsins, which are present in the eye. Opsins are apoproteins, which are responsible for vision. Rhodopsin is needed for dim light vision, and iodopsin is needed for bright light and color vision. Thiamine pyrophosphate, mentioned above, is a coenzyme derived from thiamine or vitamin B1. Thiamine pyrophosphate is a cofactor in enzymes which catalyze oxidative decarboxylation and transketolase reactions. Riboflavin, or vitamin B2, is the precursor not only for FAD, but also flavin mononucleotide, FMN. As mentioned above, FAD transfers electrons, and this is also the case for FMN. These coenzymes also carry hydrogen atoms for oxidation reactions in the citric acid cycle and the electron transport chain. Vitamin B6 gives rise to the coenzymes pyridoxal phosphate and pyridoxamine phosphate. As many as 120 enzymes require one of these enzymes as cofactors. These including decarboxylases, dehydratases, desulfidrases, racimases, synthases, and transaminases. Pyridoxal phosphate and pyridoxamine phosphate are also involved in the breakdown of amino acids, the building blocks of proteins. Vitamin B12 or cobalamin are modified to become the coenzymes methylcobalamin and deoxyadenosylcobalamin. These coenzymes are required in the enzymes which change homocysteine to methionine, an amino acid, and in the oxidation of amino acids and fatty acids. As well as this, these coenzymes assist the removal of a methyl group from methylfolate, which is needed to regenerate tetrahydrofolate, another coenzyme. Ascorbic acid, or vitamin C, is used as a cofactor in hydroxylases. One hydroxylase, which needs vitamin C, is the enzyme which catalyzes the hydroxylation of proline and lysine, two amino acids. This creates cross-links in collagen, making its structure stable. Formation of bile acids from cholesterol also requires the presence of vitamin C as a cofactor. Gamma carboxylases can require vitamin K as a cofactor. This transfers CO2 and leaves a carboxylic acid group which is able to bind calcium. Gamma carboxylases catalyze the formation of osteocalcin, which is a protein responsible for bone remodeling, as well as the formation of prothrombin, which is needed for the blood to clot. Sources: Britannica.com, pathways.org, Science Direct, CUNY.edu, and Britannica again. Further reading www.news-medical.net, also news-medical, news-medical, all of them are from News Medical. Last updated July 18, 2023. So if you're interested in the sources, the further reading, or reading this article yourself, go to the link in the description, copy and paste it, remove the spaces, and paste it into your computer or phone's address bar. That is the end of the article.